A number of methods are presently employed for genetic engineering of various animal species. Most of these were developed originally in mouse and Drosophila models, and have only more recently been extended to other domesticated animals. Access to the germline of mammals can be obtained by one of the 5 methods. First, direct manipulation of the fertilized egg, followed by its implementation into the uterus. Second, manipulation of the sperm used to generate the zygote.
Third, manipulation of early embryonic tissue in place. Fourth, the use of embryonic stem cell lines.
Fifth, manipulation of culture somatic cells whose nuclei then can be transferred into nucleated oocytes and thereby provide the genetic information required to produce a whole animal. The last 2 methods have the advantage of allowing cells containing the modification of interest to be selected prior to undertaking the expensive and lengthy process of generating animals. Usable embryonic stem cells are not available for all species of interest. However, generation of embryos by nuclear transfer from somatic cells is a method of choice for genetic duplication of genetically identical animals. 2 basic approaches presently used for inserting gene into vertebral germline cells: transfection and infection with retrovirus vectors. A third approach, based on the use of mobile genetic elements, has been commonly used for insects. Transfection methods include: first, direct microinjection of DNA into the cell nucleus. Second, by electroporation or introduction of DNA through transient pores created by controlled electrical pulses. Third, by neutralizing charges on DNA and the cell surface that prevent efficient uptake of DNA. Fourth, liposuction or lipid transfection that uses a lipid complex to deliver DNA to cells. Fifth, sperm-mediated transfection, possibly in conjunction with intracytoplasmic sperm injection or electroporation. The manner of DNA introduction is a technical issue determined empirically for each system and makes little difference to the final outcome. While transfecting culture somatic or embryonic stem cells, a selectable marker such as phosphotransferase is often included as part of the gene to indicate the success of transfection. Introduction of the DNA into the germline is a mutagenic event that will affect any gene that happens to be near the site of integration. The most obvious effect of disruption of the integrity of the gene happens in the genes that insertion occurs. Since a large fraction of the mammalian genome is non-coding DNA derived from various kinds of silence transposable elements, not all integration events will lead to gene inactivation. Direct gene introduction can lead to a number of integrated copies, its multiple sites leading to a risk of creating animals with a variety of genetic defects, which should be carefully screened in the course of subsequent breeding. For example, sheep genetically engineered for transgenic expression of growth hormone had increased incidence of reproductive problems and premature death.